will be giving a guest lecture on bioarchaeology today. So the objectives for this lecture include describing bioarchaeology in the context of biological anthropology, identifying the types of questions that we ask, and really related to this, the type of data that we can get from the bony skeleton, and then describing some of the methods we use to answer our questions. So to get us started, let's talk about bioarchaeology in the context of anthropology. Bioarchaeology defined is simply the study of human remains from archaeological sites. So this subfield really exists at the intersection of archaeology and biological anthropology. We use information from both subfields to do our studies. We really help bridge the gap between these two disciplines. Bioarchaeology is important for anthropology because we are trying to understand behavior in the past. We want to know more about the lives and livelihoods of these people that we dig up from archaeological sites. We want to know what it means to be human in the past and how that might relate to our present or even our future. So it's a very important component of biological anthropology. In terms of the kinds of things that we can learn about people using their skeleton, there are a variety of different topics I'm going to cover in this lecture. So the first of these is activity patterns. Simply enough, what were people doing in the past? If you move repeatedly in your lifetime in using and doing certain movements using certain bones, it will show up on the skeleton. And we can track that once we recover skeletal remains from archaeological populations. We can also examine diet, what people were eating, what were they regularly consuming. Migration is also detectable using the bony skeleton, so the movement of people during their lifetime from point A to point B. Health, looking at how people were healthy, looking at nutrition, things like that. Also paleodemography, the composition of past populations, looking at the relative composition in terms of number of children to adults, mortality, fertility, etc. And finally, patterns of violence, so traumatic incidences, and disease in the past. And all of these different areas of inquiry help us arrive at a better understanding of people. So let's first talk about activity patterns. Living bone can, within limits, adapt its form according to the mechanical forces regularly imposed on it. So thinking of it in a simpler fashion, bone is strengthened where it's needed, where the muscles are constantly pulling on it, and reduced where it isn't. So we can define physical behaviors that are associated with certain movements. And we do so by looking at skeletal joint variation and musculoskeletal stress markers, or MSMs. These are areas of muscle attachment on the bone that vary in terms of their appearance depending on how much that muscle was pulling on the bone. So looking at this image on the right, we have two ulnae. And you'll notice on the olecranon process there's a little bit of a difference. Ulna A has that hooked appearance, whereas ulna B does not. This is an example of a skeletal joint variation that might be related to activity pattern, wherein person A was using their elbow in a little bit of a different fashion more regularly than person B. To provide a specific example, we can look at the humeral shaft. So the musculoskeletal markers are scored at the insertion of the deltoideus muscle on the humerus, at the deltoid tuberosity. So notice on the left-hand side, we're looking at mid-shaft on the humerus. On the left-hand side, the humerus is fairly straight. There's no pronounced raised area around the deltoid tuberosity. Moving to the right, it becomes much more pronounced. much more robust. These are all humeri. And if you'll notice, I'll use my pen here, you can see in this area especially, there's a significant difference in terms of robusticity. So in the case of this particular study, males were found to have higher musculoskeletal marker scores than females. They had bigger MSMs at that site. And this can be related back to sex-based differences in movement of that humerus. And using archaeological data, bioarchaeologists can relate that back to activities, specific activities unique to that culture that maybe the men were doing at a higher prevalence than women. So you might be able to relate this back to something like hunting behavior, etc. For another case study involving musculoskeletal stress markers, let's talk about the island of Gotland. And this is a Stone Age population on the island of Gotland in the Baltic Sea. So we're talking about the Neolithic period, around 2750 to 2300 BC. And it's a marine setting. We know from the archaeological data and ethnohistoric data that these people were fishing, hunting, and gathering. And what the researchers did was they looked at 3 likely activities that the people in this population could have been doing based on archaeological remains. So they looked at archery, because they found archaeological evidence they were hunting using bows and arrows, harpooning, and kayaking. And these 3 activities use a different set of muscles for each. So researchers looked at the pattern of musculoskeletal markers in men and women, and they found that males were positively correlated with archery. So they had the musculoskeletal stress markers that were indicative of regular archery behavior, whereas that was absent on females. They related this back to a sex-based difference in archery. They also noticed the same difference in terms of harpooning. Males had positively correlated musculoskeletal stress markers with harpooning. And what's even more interesting about this harpooning argument is that it looked like there was a preference in terms of which hand the men were using to throw the harpoons and spear things, in that most looked like they were right-handed, so the musculoskeletal stress markers on the right arm were much more pronounced than those on the left. And finally, in this study, they didn't find any significant evidence for kayaking based exclusively on musculoskeletal stress markers. So you can see how we're taking raw data, raw observations on the bony skeleton, and doing some comparisons, doing some analysis to better understand the actual behavior of the people, taking the raw data a little bit further, taking it to the next step. Another important line of inquiry we can explore using bioarchaeological data is diet. So, assessing diet in the past is predicated on this statement. Basically, you are what you eat. The foods that you intake and consume show up in your physical remains, in your skeleton. And this is important for our purposes because we spend a lot of our lives eating. We need to consume food in order to survive. And as a result, a lot of our behavior revolves around procuring and consuming food. So ultimately, that Twinkie that you ate last night at midnight prepping for your finals will show up in your skeleton eventually. So how do we study diet? How do we get at this issue of what are people eating? We do so predominantly using isotopes. So we're going to talk a little bit about chemistry. Don't worry, it's chemistry light. But an isotope is an atom whose nuclei contains the same number of protons, but a different number of neutrons. And so looking at this image at the bottom of your screen, we have the 3 isotopes of hydrogen. So protium is the name for the isotope of hydrogen with just 1 proton, 0 neutrons. Deuterium and tritium have the same number of protons, but a different number of neutrons. So during chemical processes, these different isotopes, all of the same atom, behave slightly differently. The isotopes with more neutrons react more slowly than the lighter versions of these elements. As a result of this difference in reaction time, there's variation in the natural abundance of isotopes. And we see this in food items, and it tracks consistently between consumption through digestion and incorporation into the skeletal remains. Now with the isotopes, there are stable and unstable isotopes. And for reconstructing diet, we're interested in those stable isotopes that do not undergo radioactive decay. And when reconstructing diet, we use some very specific isotopes. So let's talk about those really quick. In order to describe and study the vegetable, non-meat-based component of diet, we look at stable carbon isotopes. And what we can do using stable carbon isotopes is determine the relative amount of different plant types that are being consumed by an individual. So there are 3 different signatures we can pick up using stable carbon isotopes, and these are related to differences in photosynthesis that result in different carbon values for these plants. So C3 plants are your fruits, veggies. They include things important like rice. C4 plants are a lot of weeds and grasses, and the most important C4 plant, especially when doing dietary studies in the New World, is corn. It's maize. So in the Americas, we can do some stable carbon isotope studies and look at the relative proportion of C3 plants that people were eating versus C4. And the main C4 plant that they would have consumed here is corn. So we can look at corn consumption specifically. The final type of photosynthetic pathway is CAM plants. And these include things like succulents and cacti, and they have a slightly different signature than C3 and C4 plants. So with our stable isotopic studies, we don't necessarily get results that say, okay, this person was eating this amount of corn on this day. Instead, we're looking at patterns over periods of years in an individual's life and the relative proportion overall that they were consuming of the different plant types. In order to get at protein consumption, so like meat consumption, we use stable nitrogen isotopes. And stable nitrogen isotopes track trophic levels. So if you think of something like a food chain, this is what we're tracking using stable nitrogen isotopes. So they enter the ecological system via nitrogen-fixing bacteria in the soil, which is incorporated into plants, and herbivores eat the plants, and there's a chemical reaction that occurs during digestion. Moving up the food chain, carnivores eat the herbivores, and again, you have a chemical reaction. So as a result, at these different levels, there are different nitrogen values. So we can look at the nitrogen levels in people from archaeological populations and assess how much meat they were eating on a day-to-day basis. So using this information, what can we really tell about behavior? Well, let's look at an archaeological problem and how dietary data was used to approach it. So the Maya civilization of Central America was a very complex civilization. It had the only known fully developed writing language in pre-Columbian Americas, had very complex art, architecture, math, astronomical systems, so on and so forth. In terms of time period we're talking about, the Maya originated around 2000 BC. This is when we really start seeing something that we can culturally call Maya. Although there were people in the area before that, it isn't until around 2000 that they seem to come together as this distinct cultural group. And they exist until the present day. There are plenty of TV shows and popular commentary that say, oh, the Maya are gone, there was the Maya apocalypse, whatever. They're actually still existing today in Central America. In their archaeological past, around 900 AD, something really interesting happened. So the classic period of Maya civilization was a time of extensive development and construction. It began with the first known writing and ended in an event that we call the classic Maya collapse. Suddenly, all of this complex architectural construction just stops. Writing stops at a variety of different sites in the Maya area. And for a long time, archaeologists were shaking their heads, like trying to understand why this would happen. Why is there this sudden halt around 900 AD when the civilization just seems to cave in on itself? One of the models that was originally proposed to explain this collapse is called the ecological model. And this model attributes the downfall of Maya society to an overexploitation of the environment due to problems sustaining intensive maize production in a very fragile ecosystem. So the idea here is that during the classic period, these large Maya sites were growing and growing and growing in population. You had more and more people in the same area. And as you get a growing population, you need to support them. You need to support all these different people. So the argument is that the Maya were exploiting their environment at higher and higher levels until it reached this tipping point when it could no longer support the populations and everything collapsed. So to test this, researchers used dietary data, and they developed two hypotheses. The idea was that there would be a decreased reliance on meat as meat became unavailable, overexploiting the environment, less meat, less animals to hunt. And there would be an increased reliance on maize. As dietary variation is decreasing due to this overexploitation, people were eating more maize to make up that caloric deficit. So what does the data tell us? What does the data say? One particular study was done in the Pasión region of Guatemala, which is circled here on the map. And the data examined by Dr. Lori Wright of Texas A&M University indicates that maize consumption did not universally increase with agricultural expansion. Instead, there were pretty uniform levels over time. Furthermore, animal protein supplies were not compromised by increasing population density. So possibly deforestation hadn't reached a critical point to impact the animal population, or there's something a little bit more complicated going on. So the hypotheses for the collapse were not supported by the dietary data. We can also examine migration using isotopes, for example via oxygen and strontium isotopes. And we can do this because the isotopic composition of our water and our food reflects that of the physical and climatic environments from which we're pulling the resources that we're consuming. And therefore, once we eat or drink something, that isotopic composition of what we're consuming is incorporated into our bodily tissues. So, for example, strontium in the bedrock moves into the soils and the groundwater and up through the food chain. Now, local values for strontium differ depending on many different environmental factors. So the strontium isotopic composition of human bones and teeth therefore matches the local geology. And one interesting thing that we can do using these isotopes is assess where people are born and where they die. And this is because our dental enamel, that is the white material we see on our teeth, is formed during childhood. And once you get your teeth, once they form, they don't remodel, they don't heal themselves over time. So your teeth are a permanent record of what was going on in your body during childhood. And this includes the strontium isotope values. In contrast to that, the rest of the bony skeleton is remodeled throughout the lifetime. of an individual. So the materials are turning over. And so your bony skeleton will reflect your location within the last several years of your death. So by comparing the isotopic composition of our teeth to other bony materials, we can ascertain the migration patterns of an individual during their lifetime. And to give an illustration of this, let's take a look at Teotihuacan. Teotihuacan was a ceremonial center in the Mexican highlands. It's in central Mexico, about 30 miles northeast of modern Mexico City. Now, ethnically, it's unclear who the people that built Teotihuacan really were. The earliest construction occurred around 200 BC, and at its height, it was the 6th largest city in the world, with an estimated population of over 125,000 inhabitants. So it's a very, very large center. And its influence, its political and artistic influence, it is seen throughout Mesoamerica. Around the 7th to 8th century, there was a sudden collapse, and Teotihuacan was largely abandoned at that time, and we see evidence of burning on some of the buildings. Now, it's unclear if it was attacked or if it was ceremonially abandoned, so we're not really sure about that. Modern-day excavations in the site have yielded some burials that we can do a migration study on. And a sample of some really interesting burials comes from the Temple of the Feathered Serpent. This is the 3rd largest pyramid at Teotihuacan, and it was begun being constructed from around 150 to 200 BC. And archaeologists have uncovered more than 100 possibly sacrificial burials below the pyramid itself. And this is what some of those burials look like. So here we have a group of males buried under the Temple of the Feathered Serpent. Based on their body positioning, it looks like they may have been bound at time of death. So if you look at the arms of these individuals, they're actually placed behind the body, almost as if they were tied behind these men's backs. And so archaeologists have suggested that maybe they were sacrificed. These individuals have very elaborate grave good assemblages. And one of the most fantastic items found in the grave good assemblage are located around the neck of the male burials. And so you can see here, as I move my cursor, there's this semicircle of items around this individual's neck, and you see some on the individual at the right as well. These are actually imitation and real maxillae and mandibles around their necks. So they're parts of people's mouths, or shell imitations of teeth and dental arcades. Based on the positioning of these maxillae, it appears that they were likely strung together as some sort of necklace. So, considering our isotopic information, we can ask 2 different questions of these burials. First of all, who are these men? Where are they coming from? And secondly, who are the people whose maxillae were taken and worn by these individuals? So let's address the first question. Who are these guys? Well, based on some skeletal data and the grave good assemblage, it appears that they were likely involved in warfare. So archaeologists are calling these men soldiers. Based on the dental enamel of a group of these, the soldiers themselves are largely non-local, although the exact region of origin is somewhat difficult to determine. But we can reconstruct that some of them were coming from fairly diverse regions, including Oaxaca, the Gulf of Mexico, etc. So what we can infer from this is that the Teotihuacan policing force that we're seeing, this army of soldiers, is multi-ethnic. They were taken from neighboring communities around central Mexico. Based on the bony material, the isotopic composition of the bones, they had spent a very long time in central Mexico prior to their deaths. And the hypothesis to suggest this patterning of childhood spent elsewhere and then an adulthood spent in Teotihuacan area is that they were likely taken to Teotihuacan during adolescence, possibly for some kind of military training or similar. To address the second question, who are the people represented in these necklaces of maxillae, it seems that there's a variety. Some of the teeth are foreign in nature, indicating that these soldiers were taking part in broad-scale, far-reaching military campaigns. So they were cutting out the mouths of people on their campaigns elsewhere in Mexico from pretty diverse regions. In addition, some of the teeth are very local. They have a very local isotopic signature, indicating that some of the victims were likely from Teotihuacan itself. So what we have here is a policing force both for foreign military campaigns as well as for maintaining order within the Teotihuacan center itself. So archaeologically, we can use bioarchaeological principles and isotopes in this manner to really gain a better understanding of a fairly impressive, unique burial assemblage. Analyses of bone, we can also examine things like health. And by health, what I mean is basically a general condition of an individual's body. And using bony material, we can assess health a variety of different ways. So what we're interested in here is, was someone sick a lot? Were they nutritionally stressed? So on and so forth. And we can look at things like stature. So taking measurements of long bones and reconstructing how tall people are. We can look at linear enamel hypoplasias. And these are what you're seeing on the screen in the picture. So if you take a look at that picture, you'll notice there are these linear bands that are parallel on the surface of those teeth. And what those are evidence of are stress events during childhood. So basically, when the enamel of that tooth is being deposited, a large-scale systemic stress event can cause a disruption in enamel deposition, and that causes the linear depression, the hypoplasia. We can also look for evidence of iron deficiency anemia on various locations of the skull. Dental health can be really informative to assess health overall as well. So looking for things like periodontal disease or cavities. And finally, we can look for evidence of infection on the bony skeleton and then degenerative joint disease as well. So looking for wear at those joints. And when reconstructing health, bioarchaeologists can choose to look at just one of these lines of evidence or a suite of different information to solve an archaeological problem. And one interesting question that bioarchaeologists have turned their attention to is health at the origins of agriculture. So around 10 to 12,000 years ago, we see agriculture developing in a variety of different locations around the world, somewhat independently it seems. So accepted origins of agriculture are listed in green there. So we've got the Fertile Crescent in the Middle East, China, the United States, Mesoamerica, and South America. And when originally considering agriculture, many scholars assumed that, hey, agriculture is great. And the reason for this is because the population growth after the development of agriculture has been one of the most dramatic features of human history. So originally it was postulated that this population growth resulted from improved health as a result of agricultural food surpluses. So basically the idea was that, okay, we're agricultural, we can produce a lot of food, and because we have extra food, we can support more people. And this was thought to be a very positive thing. However, reconsidering the issue, we know that agriculture is associated with increased sedentism. So people are staying in the same place to tend their fields. And with this sedentism, you get issues of communicable disease. When you get a lot of people in a small location with poor methods of waste disposal, there can be a lot of disease and it's very easy to spread. Also with agriculture, a trend that scholars have noticed is that the populations begin relying on a staple crop, so just one crop, that isn't necessarily nutritionally adequate for a healthy lifestyle. So we have two competing hypotheses here. One, agriculture was good, basically, and the counter argument is that agriculture actually had some pretty negative consequences on our species. And so what we can do is we can look at those indicators of health on the bones to try to understand this problem. And comparing populations from before agriculture to after agriculture, we see that there is no significant correlation between improved health and the development of agriculture. In fact, what we see is actually the opposite. Bony indicators of health typically suggest that there was declining health after agriculture became prevalent. So what we see is an increased number of lesions, lower stature, increased dental disease, so on and so forth, after agriculture developed. Now this could mean that health absolutely deteriorated after agriculture. However, scholars have come back with a counter-argument to this idea, and basically they say that bone takes a while to respond to any kind of stress. It's not an overnight process where, okay, you're going to get sick and then it's going to show up in your bones. That's simply not how it works. The bony skeleton is one of the body's last places to really be affected, so it takes a while for conditions to show up there. So instead, what we're seeing with this increased number of lesions after the development of agriculture could just be that people were actually surviving long enough for the bony lesions that indicate poor health to develop. Whereas before agriculture, there wasn't enough time. People weren't surviving long enough for the skeleton to really show any evidence. And this is an ongoing debate in bioarchaeology that scholars continue to discuss today. Another bioarchaeological area of inquiry is paleodemography. So we're interested in reconstructing the demography of ancient populations. So the study of ancient human mortality, fertility, and migration. So we're interested in looking at changes in pre-modern populations in order to determine something about the influences on the lifespan and health of these early peoples. So paleodemography then is just looking at the demography or the demographic composition of the ancient populations we're studying. Or alternatively, we could say it's the study of ancient human mortality, fertility, and migration. Now the way we do paleodemography is we estimate and reconstruct demographic profiles from skeletal data. And this requires good age and sex estimates. Now one of the things bioarchaeologists keep in mind when doing paleodemography is that there's an inherent problem in the sample that they're using to reconstruct these demographic profiles. And that problem is basically the idea that the skeletal population that they're using is dead. And presumably these people died for a reason. So we're not really sure if the populations are accurate representations of the demographic composition of the living population at the time. That being said, it's still a very useful pursuit, so long as we keep in mind some of the issues with it. Now, how do we actually do paleodemography using a bioarchaeological sample? So first of all, let's take a look at the Black Plague in Europe. So the Black Plague is caused by Yersinia pestis, or Y. pestis, which is a bacteria. It likely originated in China and was transmitted to Europe via India. Now the infection is transmitted to humans via fleas. And what it physically manifests as is a fairly horrible, fairly fatal condition at the time. So the Black Plague caused buboes, or these lesions, in the groin, neck, and armpits, which oozed pus and blood when they opened up. Patients had acute fevers and often vomited blood as well, and most victims died within 2 to 7 days after the initial infection, so it was highly lethal, and this is evidenced by the death toll. Estimates range from about 75 to 200 million deaths caused by the Black Plague in 14th century Europe. On average, that's about a 25 to 50% mortality rate, which is astronomical. Just think about it. Think about the population of your hometown. If suddenly an event happened or an illness happened that wiped out almost half of the population, that's fairly devastating, and we would expect to see that devastating catastrophic event using paleodemographic profiles. So what can paleodemography tell us about the Black Plague? What scholars have done is identified cemeteries in England, one of which is a Black Death cemetery, where victims of the plague were buried, and the other was a standard cemetery that was not affected by any kind of catastrophic event like a plague. And the bioarchaeologists reconstructed the paleodemographic curves, which you see here. And so we have the normal expected paleodemographic profile from the non-plague cemetery, which is the solid line. And then the plague cemetery is the dotted line. And what is on the x-axis are age categories. So we have infant, juvenile, teen, young adult, middle adult, and old adult. And the frequency that they are expected to be represented in the demographic profile is on the y-axis. So when we look at these curves and compare them, there's some very clear differences. So first of all, we notice that there's a low proportion of infants in the Black Death cemetery. And what this can be related to is the fact that fewer infants were being born to die off. Because so many adults were dying, they weren't procreating. It was a very sick time. As a result, there are few infants represented in the Black Death cemetery. There's also a fairly low proportion of teens. So these are 15 to maybe 20-something-year-olds. And these are the individuals, when you think about it, that are the most mobile in a population. They're able to care for themselves, unlike children, and they might not necessarily be held down by children or a lot of familial obligations, so they're highly mobile in a population. And it's likely that the reason they're not represented in this Black Plague cemetery is because they were moving to areas not affected by the plague. So what we've done here is we've looked at two different paleodemographic profiles to gain a better understanding not only of which people were dying, which people are represented in these samples, but also inferring reasons about the distribution of deaths in a given population. Another component of bioarchaeology that we can examine is the bioarchaeology of violence. Bioarchaeology offers empirical data that helps us examine questions about humans and our long-term relationship with violence. We see evidence of very violent behavior quite early in the archaeological record. Humans have pretty much been doing awful things to one another for a very long time. So it's a social behavior that's clearly reflected on skeletal remains. And what we can do looking at the bioarchaeology of violence is we're interested not only in ascertaining what people were experiencing on an individual level, but also looking at trauma patterns across a population at large. So an example of where we can look at this is the site of Kolha in northern Belize. So this is a terminal classic Maya site. It's pretty interesting because it's a lithic production center. And so they were producing a lot of stone tools that we see being proliferated throughout the area. Now, historically, it's important to understand that the Maya have been characterized as a fairly peaceful people, especially compared to the Aztecs of central Mexico, who we know were sacrificing people at almost an unprecedented level. So compared to this very violent northern neighbor, the Maya have always been thought to be somewhat peaceful. And the reality of the situation is that, no, they also engaged in some fairly violent behavior as well. So this is taken from the site of Kolha. This is the Kolha skull pit. It's a cache of 30 different human skulls, some of which have cranial and dental modification, suggesting potentially an elite status. And we know that this is probably a primary deposit because the skulls themselves are still largely in articulation. So the mandibles are still in place, and some of them even have vertebrae still attached, as though the heads were cut off these people and put into the pit while they were still held together by flesh. On some of these skulls, there are observable cut marks, indicating that some of the skulls may have been potentially defleshed to some extent, or at least skinned. There's also a high ratio of children. A third of these skulls, 10 out of 30, are subadults. So this was a violent act that affected not only men and women, adults, but also children. And as I mentioned, although we do see evidence of warfare and violence repeatedly in Maya epigraphy, that is the glyphs that they produced, we don't have much evidence of mass deposits of human remains in the area. So this is a unique opportunity to investigate the fact that violence did occur, and we can examine the pattern of violence, especially in this deposit, to gain a better understanding of the Maya overall. Is paleopathology. That is the study of disease on skeletal remains. So what we do to assess paleopathology is mainly we examine the gross morphology of the bone. That is, we're interested in looking for changes in shape, changes in size, if there's any obvious kind of lesion or defect like you see on the screen here. What paleopathologists need to do is give an accurate description of the condition such that it could be reconstructed later on and other people will understand what the physical manifestation of the disease actually is. Once the examination of the remains has taken place, paleopathologists will actually do a differential diagnosis like you would see on pretty much any kind of medical drama. Essentially, what we do is we go through a list of the symptoms, in this case the pattern of bony defects, and then we try to match it up with a known condition or disease that actually affects the bone. And many different diseases have similar physical manifestations on the bones, so the differential diagnosis process is very critical. Some things that we consider include level of healing, because we're interested in knowing what disease activities were actually active at death versus what had healed during the lifetime of that individual. And the pattern of healing can be very informative to understand what the life of that individual was like. And finally, in some cases, especially when you have something like mummified remains, you can actually do DNA sampling to try to identify the disease. But in some cases that's impossible to do. So we have to rely on our understanding of the gross morphology of the bony material and look at the patterning of defects on the bones. So to walk through an example of this, let's take a look at the remains on the screen. So what we have here is an 18-year-old female, and there are some defects on every single picture in this screen. So starting with the skull has been cut in half, so we're looking at it from the inside out. We're looking at the internal portion of the brain case. And what we see here, using my marker, is there's this defect right here. There's a hole in the skull, and it was caused by some sort of disease process. What's important to note here is the location. So we're on the cranial vault. It's also important to note that the lesion, the defect, actually went all the way through the cranium, all the way through the bone. That's important for understanding what caused this. Moving elsewhere in the skeleton, here we have some abnormality. This is part of the distal ulna, that is the part of your lower arm towards the wrist. And you'll see that the bone here in this area is enlarged, and there's a little hole here. So what has happened is there's a perforation into the marrow cavity inside, and this would have been very pussy and painful in life. And the bone around it is just reacting and trying to seal off that area of a problem. Moving down here, we have some ribs, and it's very clear that there's a similar process going on here that we saw in the skull. There's something basically eating away through this portion of the rib. Some sort of destructive process. And there's some evidence of healing, but only half of it maybe is healed, and there's still activity at time of death. So this was an ongoing process at the time of death in this individual. And finally, here we have the vertebrae of the spine. And you'll notice, first of all, that there's this abnormal curvature. So this person would have been hunched over permanently. And what this is caused by is, you can see right here on this one, there is a lesion forming. This destructive process that we're seeing elsewhere in the bony skeleton is also affecting the spine. So these lesions are destroying this part of the vertebrae, and that makes it structurally very weak. And so what eventually happened here for this vertebra is that this portion, this body of the vert, actually collapsed, causing this curvature. You'll notice that this portion of the vertebrae is completely destroyed. So this is the evidence before us. We have a clear pattern of a destructive process on the skeleton that's affecting a variety of different areas. So we have the skull, part of the arm, the spine, and the rib. So let's compare it to known diseases. First of all, this looks to be some sort of infectious disease. And there's lots of lesions throughout the body, so we know it's affecting the entire system. It's not something that's localized to one area. So comparing it to some known infectious diseases, let's consider leprosy, syphilis, and tuberculosis. So for leprosy, what we expect to see is the facial bones will be destroyed, as well as the hands, feet, tibiae, and fibulae. So your hands, feet, and your lower legs. That's where it's going to most commonly be seen. We don't really see that kind of destruction on the facial skeleton. Here the skull is cut in half, so I can just tell you that it's not consistent with the pattern of defects seen in leprosy. Another very common condition that we see in ancient history is syphilis. And syphilis is characterized by skeletal lesions indicative of infection. And with syphilis, sometimes there's rib involvement, occasionally, but it's really not significant. Syphilis doesn't affect the vertebrae, for the most part. And when we see it on the skull, it does actually affect the skull, but when we see it, the syphilitic lesions on the skull don't actually perforate all the way through the bone. They tend to be localized to the external portion. So this is inconsistent with syphilis. Moving to tuberculosis, also an infectious disease. Tuberculosis affects the lungs. It affects a lot of the bones in the thorax area, including the spine, the ribs. We also see it affecting the long bones of the limbs. And finally, it does affect the skull with lesions that go all the way through the bone. So based on this quick, very brief differential diagnosis, what we're seeing here, the pattern of defects, is most consistent with the diagnosis of tuberculosis. So that's just one way in which bioarchaeologists can assess paleopathology on an individual level. We're also interested in patterns of disease in skeletal populations, so bioarchaeologists would do this for an entire skeletal population and try to look at the patterning overall and understand what was going on in the population at large. So that concludes the bioarchaeology lecture. And to revisit the objectives, I described bioarchaeology in the context of biological anthropology. I identified types of questions that we ask and described some of the methods we employ using case studies from actual bioarchaeologists. I hope that this lecture was informative and provided some insight into this rapidly developing field. Thank you.